Knowledge is power. Yes, knowledge can be quite powerful, but it can also be extremely scary, which is why it's a perfectly normal response for us to be filled with anxiety, scanxiety we call it, when the time comes around for us to get our regular scans that monitor the effectiveness of our treatment and give us insight into whether our disease is spreading or not. These are naturally stress-filled times, but it's important for us to distinguish between the knowledge that the scan affords and the underlying disease itself. Putting off a scan or being reluctant to learn the results of the scan is not going to change the underlying disease. It's a bit like avoiding a photograph because you don't want to capture a particular scene. The scene is going to be what it is, photograph or no photograph. The disease is a terrible thing, but knowledge can only be good. Knowledge offers us the opportunity to make strategic decisions about how we're going to fight the disease. And most treatment decisions are very time sensitive. The sooner that we have complete information and understand that information thoroughly, the better equipped we are to take positive steps forward. Recently, I attended the International Lung Cancer Survivors Conference held in Washington, D.C. and sponsored by Longevity. In one session, Dr. Jose Pacheco, one of a handful of leading lung cancer doctors in the world that came to spend a couple of days with us to share knowledge and expertise, did a session on the basic science of lung cancer. Many people who were unable to attend that session asked if there was some way that I might record some of the knowledge and expertise that Dr. Pacheco planned to share. When I returned home and started editing some of the footage that I shot in DC, it struck me how important Dr. Pacheco's session was. Sure, he labeled it the basic science of lung cancer, and it might be relatively basic to him, but I was struck by how comprehensive his session was. The knowledge that Dr. Pacheco shared can only be good. Sure, in some cases this may lead us to outcomes that we don't like, but the knowledge better equips us to do the best we can. To that end, I've divided Dr. Pacheco's session up into 3 segments, 101, 201, and 301, and I'm sharing them here on the Living Well channel. Thank you, Dr. Pacheco, for sharing your expertise, which is so important for advocacy on behalf of ourselves and others like us. Knowledge is power. And remember that knowledge is most powerful when it's shared. So please share Living Wide with anyone who you think might benefit.
Now, the way that the EGFR inhibitors work, this is the same kind of setup as the other slide. In number 2, the EGFR inhibitor is this green triangle. It attaches to the area where ATP would normally attach to the mutated protein or receptor. And when that happens, the phosphate cannot be placed here on the protein or receptor, so it cannot be activated, and so it can't send growth signals to the cancer cell. So the way that EGFR inhibitors work, as in number 2, they basically attach to where the ATP would normally attach to the mutated receptor. So it can't attach anymore, and then it can't activate the receptor by placing phosphate on it. And so it can't send growth signals to the cell. Now, there are different reasons why EGFR inhibitors stop working. I'm going to skip through this slide because everything's actually illustrated on other slides because I don't want to go too much over. So one way we can get resistance to EGFR inhibitors, and again, this applies to HER2 inhibitors, which are in clinical trials. It applies to MET exon 14 MET inhibitors for patients with MET exon 14 skipping mutations. for EGFR exon 20 inhibitors. It applies for those situations as well. So one way you can get a mutation or genetic change within the EGFR, within the protein or receptor itself. So this area of the protein or receptor where the medication attached before, it was blue before, now it's kind of this tan color, so it's undergone a mutation or a genetic change. Most commonly in EGFR, if you've had one of the earlier EGFR inhibitors like erlotinib or afatinib or gefitinib, the most common genetic change, about 60% of the time there, is going to be T790M. To osimertinib, the most common genetic change there is going to be a C797S mutation. So when those genetic changes occur where the medication attaches, we have mutations in the additional second mutations within the EGFR protein or receptor where the medication attaches. Though they alter the protein or receptor, so now the medication, kind of these green triangles, the EGFR inhibitor, can no longer attach. But ATP can attach again. And when ATP can attach again, then the receptor can be activated again by placement of a phosphate on it and tell the cancer cell to grow. So that's one way, that's how mutations can, within a second mutation within the same receptor or protein, can cause an EGFR inhibitor to stop working, and how it can tell, how those can lead to cancer growth again. Another way is that the EGFR inhibitor can be doing its job. It can be attaching to the receptor, blocking it, working, but the cancer cell can make excess copies of another protein, such as on the left here, the MET protein or the MET protein, and it can place excess copies of that protein on the surface of the cell and basically use that to signal around the blocked EGFR receptor. But EGFR inhibitor in that case is doing its job, but it's not working anymore because the cancer has made excess copies of this MET protein, placed it on the surface of the cancer cell, and is using that now to signal around the blocked receptor. It can also do that with HER2. It can do the same thing with HER2. It can make excess copies of HER2, place it on the surface of the cancer cell, and use that to signal around the blocked EGFR receptor. And it can also do that same thing with fusions. So we've recently found that a certain percentage, it's small, of EGFR mutated lung cancers when osimertinib stops working, although it's been reported too when erlotinib and some of the other EGFR inhibitors stop working, patients can develop, the cancer can make another fusion, so the EGFR inhibitor can be doing its job. It can be attaching the EGFR protein or receptor, blocking it so it can't signal, that it can have stopped working because the cancer has made a new fusion protein, such as a RET fusion or a BRAF fusion, put that on the surface of the cancer cell, and now it uses that basically to signal around the blocked EGFR receptor. And in some of those cases, you can actually combine, for instance, they've combined RET inhibitors with EGFR inhibitors, and they've been successful. Those have been done off trial, but there will be trials coming for that group. And they haven't yet really combined effectively in patients, although they have in kind of mouse models and kind of cancer in petri dish. Looking at BRAF fusions, they haven't successfully combined something targeting those with an EGFR inhibitor for that group, but within patients, but they have kind of in the laboratory. But those will likely be coming too for patients later if they develop as a resistance mechanism.
Have you looking at how to developing an artificial signaling conduit for these problems instead of going after the inhibitors?
So the question was kind of, this is a signaling problem. There seems to be a signaling problem. Has anyone looked at developing some sort of, I guess, way you can alter kind of the signaling of the cancer cells instead of using like an inhibitor of the abnormality? So people are looking at.
When you answer that question, could you also answer a question regarding HEME binding on that, as a signaling binding?
Well, I was going to mention, and by HEM you were referring to.
Yeah, iron
No.
binding.
No. So what I was And then the second question was you mentioned something about iron binding in that. So people are looking at using things called histone deacetylase inhibitors in combination with some of these targeted therapies. Those alter certain things on the genes and kind of they try to get at the root of kind of abnormal signaling. So they target alterations that occur at the DNA level, trying to prevent kind of the abnormal protein product, which would be like a fusion or excess protein on the surface. Those really haven't been too successful so far, combining with EGFR inhibitors, but that work is ongoing. But the other thing which would be nice, which we don't know, is if we had a way to predict, because somebody brought up about combining therapies at diagnosis yesterday in one of the sessions. The only issue with that is, say you have kind of this MET that may develop, if you start osimertinib, it may develop as a resistance mechanism, MET amplification, 10 to 20% of the time, maybe slightly more. But if you don't know which patients are going to develop that by doing some sort of combination targeting MET with osimertinib at the time of diagnosis, most of the patients you're not going to be helping. You're going to be adding excess toxicity. So if we had some way to kind of predict with high reliability who was going to develop what resistance mechanisms, that would be helpful in combining therapies up front, because there's not one dominant resistance mechanism. The question about iron, I don't know how iron actually plays into that, so I wish I did. So I don't. Then the other kind of thing, the other thing that can happen, like within the cancer cell itself, within this inside space, the intracellular space, there are a bunch of other proteins floating around. Some of them, like KRAS, can become mutated. So you can have a mutation in another protein. So the EGFR inhibitor can be doing its job. It can be attaching here, blocking the EGFR receptor. Then it can stop working because another protein within this cancer cell, like KRAS or something else, has become genetically altered. And the cancer is using that to basically signal around to block the EGFR inhibitor, block the EGFR receptor. That was 10-minute warning, right?
Yeah.
And then we talked about another way we mentioned earlier, and this you can get most EGFR mutated lung cancer is adenocarcinoma, but it's also, there are rare cases of squamous reported. But most of the time it's adeno, but you can get a change due to acquisition. Most commonly it's associated with acquisition of a TP53 mutation and a retinoblastoma mutation or RB mutation. And that can cause the cancer to change its appearance under a microscope from an adenocarcinoma to small cell. That's another resistance mechanism. That requires to detect an invasive biopsy with a needle, and it requires chemotherapy usually. Now those things we talked about for EGFR activating mutations, they apply all to lung cancer with HER2 mutations, the MET exon 14 skipping mutations, and then some other mutations as well. Now, outfusions, the most common outfusion occurs when this EML4 gene is placed next to an ALK gene. So we talked about fusions occur when two genes that are normally not next to each other or placed next to each other due to some event. So most outfusions are EML4 ALK. That's the most common. There are other outfusions as well that can occur that are listed there. One outfusion has not been shown to be better than another, meaning people don't live longer if they have one outfusion as opposed to the other. They don't have better tumor shrinkage or better tumor control on an ALK inhibitor if they have one outfusion compared to another. Some people in this audience may have heard about these things called ALK variants. Has anyone heard about them? Okay, no one? Okay. They really only apply for EML4 ALK. Sometimes they'll be reported in certain reports. We don't really use them basically to decide treatment up front. when someone's newly diagnosed and treated with, say, alectinib or crizotinib, the type of ALK variant has not been associated with degree of tumor shrinkage or duration of tumor control. And it's not been associated with how long someone lives overall in really any study. What it's been shown with these ALK variants is you've had another ALK inhibitor like crizotinib or alectinib, and then you start lorlatinib. Lorlatinib works a little bit better if you have variant 3. But those people have not been shown to live longer overall, just with variant 3. Lorlatinib appears to work better for variant 3 if you've been on another ALK inhibitor and stopped working. Now, the way the ALK fusions, and this applies to ROS1 fusions too, the way they kind of signal and tell cancer to grow is you have this abnormal fusion protein that's attached to the surface of the cancer. And it's the same thing as EGFR activating mutations. It doesn't require, when that abnormal fusion protein does not require a signal from outside the cell to communicate to inside the cell to tell it to grow. Because it's placed next to this gene, which is constantly kind of turned on. It's placed next to that, so now it's also kind of constantly turned on, and so ATP now can attach to it. It can have a phosphate placed on it, be activated by placement of a phosphate onto it, and then it can tell the cancer to grow. So it's the same kind of concept as EGFR activating mutations. The abnormal fusion doesn't need a signal from outside the cancer cell to communicate the inside to tell it to grow. It can just be ATP can attach to it, a phosphate can be placed on it, and tell the cancer to grow without getting any signal from outside. And the way the ALK inhibitors work is the same. They also, here is green kind of square, the ALK inhibitor is attached to the ALK fusion protein in the area where ATP would normally attach, so it's no longer attached. And then when that happens, you can't get placement of a phosphate here, and so it cannot be activated to tell a cancer cell to grow. And this applies to all kind of fusions, like ROS1 fusions, NTRK fusions, RET fusions, the same kind of concept applies. So here, ALK mutations, when they happen, you get a genetic change, you get a change in the structure of the ALK gene, where you have that gene rearrangement or gene fusion, and that affects the structure of the protein now. And so instead of being a yellow kind of rectangle, it's a different shape now. And now since it's kind of this different shape, because it's undergone an ALK mutation, the ALK inhibitor can no longer attach. And then if the ALK inhibitor can't attach, then ATP can attach again. And then a phosphate can be placed on the fusion, and then it can tell the cancer to grow. Now the ALK mutations develop when you're diagnosed with ALK fusion positive lung cancer, you don't have an ALK mutation to start with. They develop after exposure to an ALK inhibitor. And they develop about 50% of the time if you've been on like alectinib or brigatinib, that are still non-ALK mutation mechanisms of resistance. The other way ALK inhibitors can stop working is the cancer cell can just make excess copies of the ALK fusion. So instead of having one ALK fusion on the surface, you can have a bunch of them, and there's just not enough medication to attach to and block all of them. So that's another way the cancer cell can develop resistance. And then the other way that you can develop kind of resistance to a ALK inhibitor is that the ALK inhibitor can be doing its job. It can be attaching to the ALK fusion protein here, blocking it, so it can't signal to the cancer cell to tell it to grow. But the cancer cell can either have, if you look on the left and the bottom, it can have made excess copies of another protein, like MET or HER2 or something else, placed it on the surface of the cancer, and use that to basically signal around the blocked ALK fusion protein. Or it can have developed genetic mutations or changes in other proteins within with inside the cancer, like inside the intracellular compartment, like KRAS, and use those to signal around the blocked kind of outfusion protein. For sake of time, the most common ROS1 fusion is CD74 ROS1. That's the most common. There are other ROS1 fusions. One particular type of ROS1 fusion has not really been associated with better or worse outcomes to a particular treatment. And for this, what I'll mention on, is there anyone with BRAF in here? Okay. So we don't want to forget about you guys. This is a normal kind of signaling process. If you look far on the left there, there's some sort of signal from outside the cancers, from outside this cell that's required to activate kind of this cascade of signaling from RAS to RAF to MEK to ERK, and then to tell something to grow. And that's dependent on getting a signal from outside the cell. And these are all kind of actually similar concepts if you think about the alteration really leads to not needing a signal from outside the cell and kind of constant signaling. So here you have like a BRAF V600E mutation. And this will be kind of our last slide. BRAF V600E mutation, you have that, and basically you now no longer need a signal from outside a cancer cell to tell something to happen or send growth signals to the inside. This is kind of constantly turned on by this mutation, and then constantly activates all these other guys to grow, to send growth signals. The way that the medications work, lidografinib and trametinib, which people take, is that they block these two components of this signaling cascade, so hopefully it doesn't tell the cancer cell to grow. Okay, and I had a lot more slides, but we just kind of ran out of time, but that's fine. Any questions? I think I'll show one slide at the end, because Katie might get mad at me if I don't. Well, I'll do this one real quick. So the cancer cell there on the left has a bunch of immune cells around it. That's needed for immunotherapy to work. One of the other reasons, besides low amounts of mutations, why immunotherapy by itself doesn't work for kind of EGFR activating mutations, Outfusions, ROS1 fusions, RET fusions, is because besides low mutation amounts, they often have a lot lower amounts of immune cells around the tumor. And so if you don't have immune cells around the tumor, immunotherapy by itself is not going to work. Now, I think I wanted to show this because Katie might get mad if I don't. So I built that. It took a few hours. And he likes it, so.
Thank you.
Okay.
It's Living Wide's mission to help everyone lead lives filled with meaning and purpose, despite the uncertainty of life-limiting illness. Thanks for stopping by. Hit that like button and subscribe if you haven't already. And share this with friends. Literally, the more engagement we can get here on Living Wide's channel, the more people will be able to help. Thank you for being a part of what we're trying to do. More soon.